This lecture is an overview of bacterial metabolism. I like to show this regulatory decision-making tree that microbes will follow when deciding whether or not to turn on gene cascades and that encode specific kinds of enzymes. So over here on the right are the decisions made for whether or not to turn on operons or genes encoding biosynthetic enzymes. So the first question, and only question really, is the end product of the pathway present? And if the end product is not present, then the microbe will synthesize enzymes to produce that end product, and therefore genes and operons will be switched on. However, if the end product is present, and I should point out, not just in present, but in sufficient quantities so that it's not, in excess, the operons are turned off, and therefore the microbe does not synthesize the enzymes to produce this particular compound. Obviously, sometimes these things, like vitamins and amino acids and so forth, sometimes they can be toxic. And so it behooves the microbe not to make too much of an end product. But obviously, the biggest issue is energy production. You don't want to be wasting energy making something that you have more than enough of. The catabolic enzymes have a little bit more sophisticated decision making to go into with them. The first question then is the substrate of the pathway present? And if the answer is no, then obviously the microbe will not synthesize the enzymes necessary to eat that particular substrate. Obviously, if the substrate's not there, there's nothing to eat. There's no need to produce enzymes to break it down. However, if the substrate is present, there's another level of questioning here. That is the preferred carbon and energy source present? So if the preferred carbon source isn't present, and it's just this other thing, then the microbe will synthesize enzymes to eat that particular less preferred substrate. But if the preferred substrate is present, then the microbe will shut off enzymes and so forth for the degradation of a less preferred substrate, so that it can then eat the preferred substrate. I'll give you some examples of that down the road, of microbes making those types of decisions depending on the kinds of sugars that are present. Some of the major carbohydrate pathways are listed here. The Ebden-Meyerhoff-Parnas pathway, pentose phosphate, and Entner-Duderoff. And so the first set of reactions, glucose to glyceraldehyde 3-phosphate, are different among these three. But then the last phase here, the glyceraldehyde 3-phosphate to pyruvate, is the same among these three. So this is where the difference lies, and you'll see that in the coming diagrams. The Ebden-Meyerhoff-Parnas pathway, or glycolysis, occurs in the cytoplasmic matrix of most microbes. The most common pathway for glucose degradation to pyruvate in stage 2 of aerobic respiration, and it is oxygen independent, and again it occurs in 2 phases, as I'll show you. So there's a 6-carbon phase and then the 3-carbon phase that you see here. And so here you're seeing the addition of phosphates to quote-unquote prime the pump. So a microbe can catabolize glucose here, phosphorylate it to glucose 6-phosphate. And as you see, here's each of the carbons, and here's where the phosphate is added. It's isomerized to fructose 1,6-phosphate, and then an additional phosphate has to be added. So you need to use 2 energy molecules to initiate this process in order to get the fructose 1,6-bisphosphate. And then an aldolase will cut that into 2 3-carbon phosphates that you see here. And then each of these 3-carbon phosphates is then subsequently phosphorylated and in the process oxidized as well, to ultimately produce the 3-carbon pyruvate. So there's a couple important things. There's oxidation steps to generate reducing power. And again, these NADHs that you see here are then shunted off to electron transport for the microbe to produce energy by oxidative phosphorylation. In these steps here, the so-called substrate level phosphorylation events, that happen here, 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 and here are essential for making energy. So we make 4 ATP molecules and consume 2, and therefore the net overall production is 2 ATP per glucose molecule. This slide just shows you that glycolysis is actually amphibolic, so it's both anabolic and catabolic. Obviously, it's catabolic because we start with this big molecule and we end up with 2 3-carbon breakdown products of that. But it shows you this versatility of the pathway here, in that you can remove substrates at any point and make things with them. So the glucose that's here can be used to make polysaccharides needed for biofilm formation or whatever kind of mucilages microbes might make. Sometimes microbes will need extra amino acids and therefore, or amino sugars rather, to produce peptidoglycan and so forth, and so they can pull that material from here. Phospholipids obviously are necessary for making cell membranes and other kinds of vacuolar membranes. And so again, that can come from here, from the dihydroxyacetone phosphate. The microbes can also pull substrates for amino acid biosynthesis, such as serine and glycine and cysteine from phosphoglyceraldehyde. And again, here's another spot in which some amino acids, more complex aromatic amino acids, can be made, or even uramic acid, which can be used for peptidoglycan synthesis. So, and also what they show you here is this concept of feedback inhibition, where substrates can feed back and either block or keep the process rolling. And so in this particular case, there's examples of different substrates that can interfere with the enzymes responsible for these particular steps, like fructose bisphosphate in excess accelerates this conversion of PEP to pyruvate, essentially the dephosphorylation of PEP to pyruvate. Here you can see that AMP, adenosine monophosphate, interferes with fructose 1,6-bisphosphate phosphatase, so an enzyme that would pluck phosphates off this and send this guy backwards, can be interfered with by this particular substrate and so forth. So the Entner-Duderoff pathway is typically used by soil bacteria and a few gram-negatives like E. coli. It replaces the first phase of the Embden-Meyerhof glycolytic pathway. And so that's pictured here. It's a pretty versatile pathway, and again, the first stage here, the 6-carbon stage, is very different. But then the 3-carbon phase, these steps here, are identical to glycolysis. And so again, glucose is phosphorylated to glucose 6-phosphate, and then oxidized to 6-phosphogluconolactone, and then there's a hydration step, and then dehydration step to make 6-phosphogluconate, and then this intermediate KDPG, which is keto-deoxyphosphogluconate, KDPG here, is cleaved by this aldolase. So this dehydration step right here is facilitated by this dehydratase. And so now you have this 6-carbon KDPG sugar, which can be chopped by this aldolase into pyruvate, so 3-carbon pyruvate, and then the other part has to be converted to pyruvate through the typical 3-carbon reactions of glycolysis. And here's where we get our substrate level phosphorylation events. And so again, you're using 1 ATP in order to make 2 ATP. So it's not quite as energy efficient in the end. But one of the great things about this is its versatility. So you can use gluconate. So the microbe can use gluconate, for example, which is pretty abundant in fruits and things like that. So it's common, it's fairly common in the soil, and therefore soil microbes can absorb gluconate and phosphorylate it using an enzyme glucokinase to 6-phosphogluconate, and then dehydrate that and then make KDPG, and then you get the 2 pyruvates. Over here is just showing you a transport pathway that the microbes use, this proton symport, as it's called, where the microbes can transport glucanase across the permease enzyme using proton motive force from their electron transport chain. So they're pumping protons and then those protons can come back into the cell through permeases and use that to transport nutrients. So this table just shows you the distribution of EMP and ED pathways. And so again, here's a whole bunch of different kinds of microbes, and these guys are primarily soil microbes: Pseudomonas, Rhizobium, Xanthomonas, and Thiobacillus. And none of them have EMP enzymes. They're all operating through the ED pathway. And then there's a variety of other kinds of microbes. If you can focus on, like, for example, E. coli has the EMP pathway and bits of the Entner-Duderoff pathway. And so again, the idea is that some microbes are a little bit more versatile and can do some portions of these pathways, like E. coli. So that's why it says here, when organisms such as E. coli are growing on gluconate, they can synthesize the enzymes of the ED pathway, but generally don't use them unless there's a weird circumstance like this, where the microbe finds itself with an abundance of the substrate. The pentose phosphate pathway, it's also called the hexose monophosphate pathway or hexose monophosphate shunt. It can operate at the same time as the glycolytic pathway or the ED pathway. So microbes can use both of these, pentose phosphate and combination with glycolysis or ED. It's oxygen independent, and it's also amphibolic, as pretty much all of these are, meaning that it's both catabolic and anabolic, depending on the microbe's needs, as you'll see here. So the pathway looks very similar to the first stages of the Entner-Duderoff pathway. You have glucose 6-phosphate being oxidized in order to make reducing power in the form of NADPH, and then eventually you'll make some 6-phosphogluconolactone in here, which they don't show, and then the 6-phosphogluconate. So these G6P dehydrogenases are operating in here to again pull electrons off of here and make this reducing power. There's a gluconolactonase that's in here as well that helps make this substrate, the 6-phosphogluconate. And then basically, again, this thing is oxidized to make this reducing power that you see here, another NADPH. And so again, these NADPHs can be used for biosynthesis. They can be used as an electron source for biosynthesis. Ultimately, you make ribulose 5-phosphate, a 5-carbon molecule, and then a RUMP epimerase. The epimerase here will make these different side products, basically. And so again, one of them is just obviously itself ribose 5-phosphate from the ribulose 5-phosphate, and another one of these is xylulose. And so, as you see here, and then over here is xylulose phosphate. But again, these are the same thing. So again, these two here can be grabbed onto by an enzyme transketolase, and transketolase will grab 2 carbons from ribose 5-phosphate, making glyceraldehyde 3-phosphate, so a 3-carbon, and then stick those 2 carbons over here on the xylulose to make the acetoheptulose. So again, this 7-carbon. So that's basically all the transketolase is doing, is grabbing these 2 carbons and moving them over here. That's where the trans comes in. And then the next enzyme, the transaldolase enzyme, will grab 3 carbons here and stick them onto glyceraldehyde 3-phosphate. So now we make the 6-carbon fructose 6-phosphate, and then 7 minus 3, so we end up with the 4-carbon erythrose 4-phosphate. And that can combine with some of these xyluloses that are the result of the epimerase here. And then again, you have another transketolase reaction where you can grab 2 of these carbons and stick them onto here to make the 6-carbon erythrose, and leaving behind the 3-carbon glyceraldehyde phosphate. And then these are the standard EMP reactions going from glyceraldehyde 3-phosphate to pyruvate. And then some of this can feed backwards to regenerate the glucose 6-phosphate that's necessary to keep this rolling. And so again, some of the neat things about the pathway is that you're making pentose phosphates that are essential for making nucleotides. As you know, ribose sugars are a primary core molecule in nucleotides, RNA and DNA bases, and so that's where a lot of the ribose comes from. Erythrose phosphate can be used for amino acid synthesis. This guy here, and you're getting this NADPH, this form of reducing power that's necessary for reductive biosynthesis of fatty acids, sterols. So again, it's used for biosynthesis. And then there's a bit of ATP that's formed here in these reactions. So it's a very versatile type of a pathway. So moving away from carbon breakdown, now we're looking at just a little bit of a snapshot of electron transport chains. And this is just some the aerobic respiratory chains in E. coli. Not the whole thing, obviously, but just the basic pathway. So you start with some kind of dehydrogenase that would grab electrons from these electron carriers here. So NADH or FADH2 made from Krebs cycle. And these electrons would then be passed to quinones, and then ultimately from quinones to these cytochromes. So cytochrome BO is used by E. coli when it's growing under high oxygen concentrations. So this particular enzyme will be more active in transferring electrons to oxygen when oxygen levels are high. Cytochrome BD, however, is used by E. coli when the microbe is under lower oxygen conditions. So it's pretty clear that this cytochrome has a much higher affinity for oxygen, and therefore you may need that extra affinity in order to get smaller amounts of oxygen pulled out of the environment. And so this cytochrome BD will then transfer electrons to oxygen in order to keep the electron transport chain moving. So again, it shows you the versatility of a microbe like E. coli, being able to use different cytochromes depending on oxygen concentrations. Another highly versatile microbe is, I skipped ahead of myself. This is the anaerobic respiratory chain. So again, not only can E. coli distinguish between levels of oxygen, it can also deal with no oxygen and can activate anaerobic respiratory chains. So E. coli is extremely versatile. We know it's a facultative anaerobe essentially. So when it's growing in the presence of oxygen, it's most happy, but it can certainly switch gears if the right electron acceptors are present. And so that's what's pictured here. So again, the dehydrogenase pulls electrons from electron carriers to the quinones, and then the quinones can, depending on which electron carriers are present, so if fumarate is present in the microbe's environment, then it will produce a fumarate reductase. And then, so that will then grab electrons from here and transfer those electrons to a variety of carriers, and then ultimately to fumarate as the terminal electron acceptor. Sometimes E. coli can find itself in the presence of DMSO, dimethyl sulfoxide, in its environment. And so that has a DMSO reductase that can grab electrons from here and transfer those electrons from through the electron transport chain, ultimately using DMSO reductase to DMSO. And then TMNO, TMNO, and nitrate reductases as well are present. So the microbe can use TMANO or nitrate as a terminal electron acceptor. So again, that's not all. I mean, these are just for simplicity in this particular diagram, but just showing you how versatile the microbe is. If oxygen isn't present, the E. coli can use a variety of electron sources. And so, using nitrate as a terminal electron acceptor is relatively common in the microbial world. And Paracoccus denitricans, as the species name suggests, is a good example of that. So Paracoccus has both an aerobic and anaerobic electron transport chain capability. And so the microbe has a dehydrogenase that can pull electrons off of electron carriers and pass them through quinones to cytochromes and ultimately to oxygen. The microbe can also pull electrons off of things from the environment, like simple carbon molecules like methanol, taking electrons from them and passing those to cytochromes as well, and ultimately to oxygen. So again, it's a versatile microbe that can use a variety of substrates, including simple carbons like methanol. But if there's no oxygen present, the microbe can also utilize various versions of nitrate. So nitrate, nitrite, nitric oxide, and nitrous oxide, N2O. So in this particular case, the nitrate reductase will allow the microbe to do this. And hopefully you remember this from like Micro 224, where we did all these nitrate broths and those types of experiments with the addition of sulfamic acid and zinc dust. If you remember back to all that, this is basically what we were doing, is looking at these different ways in which microbes could denitrify. And so nitrate reductase, again, is essential to transfer electrons to this terminal electron acceptor. Nitrite reductase grabs electrons and transfers them to nitrite, and nitric oxide reductase, again, and so forth, nitrous oxide reductase. And ultimately, the microbes will make nitrogen as a waste product, and then the nitrogen gas can diffuse into the atmosphere and into the environment, where plants could use it to fix nitrogen, if they're capable of nitrogen fixation. Obviously, most plants would prefer to eat this. And so these guys can actually be at odds with plants in the environment, because microbes and plants can be competing for this. But again, the production of nitrogen might help certain kinds of plants with the capacity to fix nitrogen. That is plants that have nitrogen fixing microbes associated with their roots. Another really cool example of electron transport occurs in certain kinds of microbes that are able to transfer electrons to external iron oxides, and that's pictured here. And so here's the electron transport chain, and here's the cytoplasm. So here's some sort of a reducing power in the form of NADH that's generated from metabolism, donating its electrons to this complex NADH dehydrogenase in the membrane. There's quinones that you see here that pass through this periplasmic protein MAC-A, and then ultimately MAC-A will deliver electrons through this pool of periplasmic cytochromes. And then the periplasmic cytochromes can dump their electrons into these outer membrane proteins, these OMC proteins, and then ultimately up these pillin proteins, which act essentially like a wire, which is really impressive. And then from these pillins that stick to iron oxides in the environment, the iron can become, these oxidized forms of iron can become electron acceptors. And so this process here would facilitate the microbe's ability to metabolize and make energy using iron from the environment. And that's what you see here, these globs that the arrows are pointing to, illustrating this conglomeration of iron oxides that these microbes are using as electron acceptors. Again, essentially creating a copper wire. Obviously, it's not made out of copper, but another version of metabolism occurs in these photosynthetic type bacteria. And so here's Anabina, that cyanobacterium, and you see this guy under the multicellular type of microbe that has vegetative cells and then intermittent heterocysts that you see here. These are the sites of nitrogen fixation, and then the vegetative cells are capable of photosynthesis and carbon fixation. And so in this particular case, these vegetative cells, so that's here, and that's this, and then here's the heterocyst, has both photosystems 1 and 2. And so these can absorb light. They're oxygenic photosynthesis, and so they make oxygen in these vegetative cells. They also make a ton of ATP and NADPH for reducing power. They're using water as the electron donor and then pumping out oxygen, as I showed you. And then the energy and reducing power that the microbes are making is powering the Calvin cycle. Calvin cycle is basically just carbon fixation, where the microbes will take CO2 from the environment and using the energy and reducing power will turn that into organic carbon. And then the organic carbon can be metabolized here to make energy, but also can seep into the heterocyst and be oxidized by pentose phosphate or glycolysis. And that creates electron transport. And those electrons, those reducing power that's generated, can be used to fire up this enzyme, nitrogenase. The nitrogenase will, it also not only does it need reducing power, but it also needs energy. And so I should point out that the heterocysts only have PS1, photosystem 1, which is not oxygen generating. And as a result, you can generate ATP without oxygen. And that's important because nitrogenase is extremely oxygen sensitive. And so keeping this oxygen separate from this heterocyst is vital to allowing the microbe to ultimately kick on its nitrogenase, make organic forms of nitrogen. Here's this ammonia that can then be used to make amino acids. And then the amino acids can be pumped into the neighboring vegetative cells in order to give them nitrogen, to feed them essentially.